FDA here to give us an introduction to the DILI contrast data set. And oh, you're talking. All right, great, Weida. Perfect. Yes, no. It's all good. I got confused. Yeah. Okay. So
somehow I'm being removed from the presentation?
No, you're not being removed. I'm just being confused. That rhymes. All right. Well, Weida Tong is the director of the bioinformatics and biostatistics division of NCTR, and he will give us an introduction to basically a challenge that builds right onto these key questions that we learned about in the keynote. How do we make sure that drugs are safe? And it's with great pleasure to have you here, and thank you so much for starting this first challenge. Thank you so much.
Thanks, David. So sort of like a continued drug safety theme, as Larry mentioned, and it's very critical. Actually, some weeks ago, I read a paper in the Drug Discovery Review and talk about what is the best way to develop a drugs and bring a drug to the market. And there is two classes of the drugs, one called first-in-class, second called the best-in-class. The first-in-class, as the name suggested, the drug never been on the market before, is brand new. And the best-in-class, that means there is already have a lot of the drugs, the similar drugs on the market, but you came out the best. So the pharmaceutical company put a lot of the discussion, what is the best strategy to bring a drug to the market? And turns out they feel the best in the class is the best strategy. Now, what means the best? It's the safety. Basically, it's called safest drugs in this class. So among the safety, actually there's two major reasons for the drug withdraw from the market. And one of them is a drug-induced liver injury. So this is the one we're going to talk about today. So, and what I'm going to do, I'm just going to talk a little bit about the drug-induced liver injury in case you are not in this particular area. And we're going to talk about why we care and why we want to use the genomics data. And lastly, I'm going to turn to the Dr. Shraddha Thakkar, and she will explain what this project looks like. Okay? So what is the drug-induced liver injury? Basically, injury in the liver by taking the drugs. So when you take the drugs, and the first sign of the liver injury, and you will see an enzyme called ALT and elevated. That means your liver got a punch and release the enzyme to the bloodstream. You will see the elevated ALT. And you go to the routine physical exam, is what I did. And I took a lot of alcohol before I go to the doctor, so that's why the ALT got elevated. And the doctor does not really take it too much serious when you see the ALT elevated, because there is many reasons to cause the ALT elevation. For example, if you fell off the stairs, you will see the ALT elevated. Okay, and besides, the liver is absolutely strongest organs in your body, and you cut in half, another half going to grow back. So, doctor just going to tell you, okay, waiting for 2 weeks, come back. Let's just see the ALT going to come back, right? And then after 2 weeks, you come back. Of course, you are not going to fall off the stairs every day. So if ALT is still elevated, that means your liver really have a problem. But that might be okay. The really worrisome is another enzyme starting to kick in called the total bilirubin. Okay? This enzyme measures the function of the liver. When this enzyme popped up, that means your liver really in troubles. Right? So at that times, nothing you can do about it, and the only thing is liver transplantations. Well, there is not many liver lying along, so that's why you go to the driver license, they ask you for the organ donor, and that means if someone hit by the car, your organ going to be taken away and used for that kind of the purpose. So a lot of the times, the acute liver failure going to cause the death simply because we do not have too much liver around. So in this field, there are two major questions, right? The first question is more on the clinical space. And when the ALT elevated, we're going to ask whether we will be able to know total bilirubin going to follow. And there is no biomarkers available in the clinical space. And if we don't do nothing, let's just say you have 5 patients walking to the hospitals, exactly same symptoms, all the ALT was elevated like hell, you do nothing about it, 4 will walk out the hospital, it's okay. The liver just that strong. Another one probably going to need the liver transplantations, but we don't know who going to be safe, who will be not. So this is the biggest challenge in the clinical space. Another one is more on the drug development. And this is what this particular project is for. So in the drug development space, and we wanted to come up a drug not to cause harm to the patients, and particularly drug-induced liver injury. Okay? So why we care? Why this is important? Well, I can give you a long list of the things you should care, but I just want to highlight one thing. Around 40% of the drugs failed in the clinical trial or the post-market surveillance due to the liver injury was not detected by the preclinical study, particularly the animal models. That really means whatever the model we have to assess the safety in the preclinical space is not very good, right? So that's why there is a lot of the study in this space, particularly using the genomics, in silico approach, and in vitro assays. Those sort of the approach constitutes so-called the 21st century toxicology. And the 21st century and toxicology has been endorsed by the research community and particularly the government agency. And in the FDA, we released advanced regulatory science, the white papers, and the encouraged community came up good methodologies and using this technology to assess the drug safety. And we heard about the TOX21 and TOXCAST program. Actually, TOX21 is the interagency collaborations between the FDA, NIH, EPA, and the National Toxicology Program. And we have a TOXCAST program. This is EPA-centric. They are more interesting on the industrial chemicals. And all of these programs have a strong component of the genomics methodologies. You might heard about and the EU REACH program, actually the EU REACH and the registrations, and emphasized the 3R principles. That means to replace, reduce, and refine animal models. Basically what they say is get rid of the animal using the in vitro or genomics technologies. So in this particular domain, and you can see there's a lot of the genomics data already available. And this is one of the probably the largest data set in this field. It's called OpenTG Gates. Actually was developed by a Japanese consortium activities. They put $50 million on it and work on 10 years and generate a humongous database. And I put some of the data in the previous CAMDA challenge, and it was just fantastic. We see so many wonderful analysis take advantage of this data set. Let me just spend a few minutes just explain to you about this particular data set. On this site, there is 4 testing systems. On these 4, it's routinely used in the preclinical space. Okay, first is in vitro assays. We can use rat primary hepatocyte, basically fresh out of the liver, or the human primary hepatocytes, okay? And we treat it in the in vitro, and then 2 hours we pick the samples, or the 8 hours or 24 hours, so we can take the sample in the multiple time points. And then we can do the in vivo assays, and this is we just give a rat a strong dose, and 3 hours later we pick the sample, or 6 hours, 9 hours, or 24 hours. So you can do that, called a single dosing strategy. And the most routinely used on the preclinical models in the pharmaceutical company is it's a repeated dose. That means we repeatedly treat the rats with that chemicals for a certain amount of days, and then we took the samples. And one of the typical On the preclinical study, it's 28 days and studies, right? So what the Japanese group does is they look at 170 compounds, most of the drugs, and look at the control samples, look at the lower dose, medium dose, and high dose, generate a huge amount of the data. So we put this data out, how many years ago? 3 years ago or 4 years ago. So we run the CAMDA challenge on it. Now, there is a couple of issues. First, let's just say, be clear. We are trying to work away with the animals. So for this year, we are not talking about the data from the animal studies. We are talking about in vitro assay. So rat primary hepatocyte data does not resemble very well to predict the liver injury in humans. So this is pretty much out. And the best in vitro systems actually using the primary hepatocyte from human. Well, there is a problem because when you do that, first of all, there is a huge variability between the individuals. Let's just say you do the experiment today using the one donor and the female, 56 years old. Unfortunately, she was hit by the car. Now liver is available. We took the primary hepatocyte to do this experiment, but somehow the samples run out. Next person, maybe it's the male, and there'll be very different physiology. So the results is very difficult to control. So the best way to do is use the immortal cell lines, such as the breast cancer cell line, prostate cancer cell line. Those cell lines has been routinely used and in the various settings to screening the drugs and particularly for the efficacy. Why we cannot use for the safety assessment. So, and the question when we propose that, the question always asks, well, what the breast cancer cell line got to do with the liver? Because they're not even in the same organs, right? Is that really relevant? Actually, there is some studies. I just listed one publications in the Nature Communications on the last year, indicating actually breast cancer cell line is not bad to predict the drug-induced liver injury. And of course, there is a lot of the advantage to use the immortal cell lines. That's no question about it. First of all, endless amount, and we can do a lot of the analysis. And the most importantly, a lot of the data we needed already been generated. We don't need to generate any data. So the objective of this year's CAMDA project is to understand whether the MCF7 cell line and PC3, that's the prostate cancer cell lines, the genomics data from the CMap, will be able to predict drug-induced liver injury and in humans. So with that, I'm going to invite Dr. Shraddha Thakkar, and she actually was the lead of this project. She's going to tell you a little bit more about the study design. And
I'll just take it from here.
You want it?
Am I audible? Okay. So moving on to the just basic study design, many of you in the audience participated in this challenge. So what we did this year, just to use the CMap data, and you must be dealing with this data if you have submitted your model to us. So the idea behind is to get the data and get different models from CMap data, which is the whole genome array, and we are planning to compare, contrast, and combine. So the results which will, individual models will be discussed by presenters who submitted the data to us. At the end, we are just going to talk about compare and the contrast, but we have not done the combined analysis, and at the end of the meta-analysis presentation, we're going to dwell into that particular category. But the idea is to compare, contrast, and combine different models to understand, can CMap whole genome array using MCF7 cell lines or PC3 cell lines can predict drug-induced liver injury. So we want to understand the strength of different methodology, because everyone using the same data but developing different set of model, different variety of model, different strategies, and also using either can be model effective individually or in combination. So what we did, these two cell lines from CMap, and we just have only single objective from the study is just, can we capture DILI signature effectively from these cancer cell lines. So for this study, we have 280 drugs in whole training and validation set. Now, we assign them as a DILI positive and negative, and these are the training set which was given with the class label. Now, if you heard Dr. Lasko's presentation about real-world data, and that is the information we used to classify these drugs into positive and negative, and that's the strategy we are trying to implement, bottoms-up approach, where we use the human real-world information, implement it with clinical data, and see can we have the predictive analysis. And then he actually mentioned that we are very safest in that category. The results are very predictable. So we are trying to see can we use the cell lines because we are strongest in this area right now. And also there are 86 drugs given to everyone blinded. They did not know anything about it. And people developed the model, freeze the model, gave it to us, and also gave us the prediction of those 86 drugs. But we knew internally what they are actually. So now, so just to tell you a little bit about the data, because nothing was released with the data set. They all were the therapeutic dose, only one single data point, means 6 hours time point. All the data sets were generated with one vehicle DMSO, and there were 2 arrays used in the data set because there are some drugs which were developed using another array, kind of. So just give you overview how long it take us to put together. I'm not even counting the time which we spent before we announced this challenge. So we announced this challenge to everyone with help of David and Pavel in October, and of course we give them a lot of trouble by putting the data together in few months. But anyway, we released the data set by December, and then within short time, I really want to acknowledge everyone's effort to put together these wonderful models and provide it to us by March and April. I mean, it's a commendable job to deal with this such a complicated endpoint and such a complicated data and give us the model by that time. So by April, we had freeze model with us. And then everyone developed their extended abstracts by May, and now we are here to discuss about top performing or better models, and also the meta-analysis which we conducted based on the freeze model and predictions given to us. So just to give you an overview, what all we have so far, we have around 10 data analysis teams. Actually, one dropped out from Asia, so we have total 9 so far. So some are from Europe, some are from US. So we have different variety in demographics as well. Now, just to talk about the study design, we gave 2 sets. So one is the training set which people use to build the model with 190 drugs, and the DILI class labels were provided to them. However, validation set was just blinded. They did not know anything about it, and the same data set was available for PC3 and MCF cell lines, single dose, single time point, and it was provided to participants to develop the model. So model was developed individually by individual participants, and we asked them to freeze the model. So once they freeze the model, everyone freeze their model, submitted the predictions and the results from the freeze model to us for the meta-analysis. Once we received that information, we provided the DILI class label information to each participant. So then they can look it up and see how their model performed. And with the same time, we started analyzing different models and different predictions, what we have, to see how is this scoring happen, how is the prediction happen. And now we will be sharing tomorrow the part of meta-analysis. And also we have very exciting results through this meta-analysis. Also, it going to continue into further challenge. So please tune in for entire meeting and then be with us tomorrow to see what's going to lead to that. Now, just give you a hint that there are multiple teams, multiple crazy strategies, multiple analysis. Even we were astonished by the variety of information we received. So we are sitting on the wealth of data, like Larry said, and we really want to see how we better analyze it, how we better predict it, and we are in right now safest zone because this is our strongest point in terms of data prediction. So tomorrow we'll be comparing and contrasting, but however, I would need your engagement further including whoever participated in this challenge, to have the combination efforts so that we can have better predictability by combining the model. So with that said, I just want to thank you all for submitting us to participating in this challenge. Big thank you to David and Pavel to help us conduct this consortium effort. Of course, spearheading with me, Dr. Weida Tong. We had hours of discussion finalizing this. However, this is the small component of big project we are running in FDA, and that many of the people are sitting in the audience gave us very good insight and advice for planning this project. Like Dr. Lasko, he gave us billions of advice before conceptualizing this project. We also announced this project to MAQC Society. Some of the people are sitting in the audience. They submitted excellent models to us, and I'm really thankful to have this support. In addition to that, we are going to lead towards another talk, which is going to talk about the models we developed in-house. I just want to clarify that the model we developed in-house has the same level of stringency which everyone had it. It was blinded, not given any information, even though he's my colleague and friend. I did not tell him anything, and treated as the same as different participants. And thank you so much for your time. I'm looking forward for excellent presentations.
Thank you very much. Yeah, we should very much hope so that no one gets to see the blinded data. That's the whole point of the contest. So thank you so much for introducing this exciting challenge. We have time for one or two short questions regarding the nature of the data set, and then we will move into the presentations that analyze it. Yes, please. Can you come to the microphone, please?
Thank you. That's a very interesting initiative. I just wondered why you left out the HL60 cell line, which is also in CMap.
Oh, yeah. So we are trying to see can we cover the maximum number of drugs to get a real signal out of it, and that's what we are trying to move towards the L1000, which we are going to talk about it next year. So HL160, it was not giving us enough coverage, and we want to have equal number for have better comparison. So once we create the platform for the comparison, that's why we want to move on to the cheaper method to identify drug-induced liver injury. So we are planning to move to L1000, include more cell lines so we have better analysis. Yeah, that's the teaser for tomorrow. Thanks.
Thank you very much. Any other questions? Yes, please.
Can I ask a question? Yeah. With the MCF7 lines, we frequently will take anti-cancer drugs and put them in there and look at the cell viability and activity of those. Is there any relationship between if I had a strong activity in MCF7 cell lines, say from cyclophosphamide, and drug-induced liver disease? Are the two correlated at all?
No, actually, not at all. Like yeah, pharmacologically, not at all. But maybe, we don't know. Yeah, we are just working on it. But yeah, pharmacologically, no correlation at all.
Okay, so thank you very much. It's my pleasure to